So I welcome everyone to join our September ML Plus X forum. For those who are new to this event, this is a monthly forum in which we will have a few sort of presentations to highlight the certain aspects of machine learning and its applications. So today, we're going to have three presentations centered around this topic called multimodal machine learning. Before we move to our actual talks, I'd like to take this opportunity to thank our sponsors who have been very supportive for our community and our events. So if you're sitting here and you're interested in sponsoring us, feel free to click at that link that will direct you to the web page with all the information for sponsorship. And finally, a very quick reminder that we will have the ML Plus Coffee event tomorrow morning, scheduled at 9 to 11 a.m. So those will be the events that you can grab some free coffee and talk to your peers and our community members about anything about machine learning. So if you're interested, it's going to be in the same building on the first floor. It's going to be the Harbor Central lobby on the first floor. And do bring your laptop so that you can have a discussion and show something on the screen. Now, with that, I would like to have a quick introduction of our first speaker and also my colleague, Daifeng Wang. So Daifeng is currently Associate Professor in Biostats and Microinformatics Department and also affiliate faculty member in Computer Sciences. And his research focuses on developing machine learning and bioinformatics tools to analyze multimodal data for complex brain and brain disorders in order to understand the underlying cellular and molecular mechanisms. Without further ado, I'm going to hand over Daifeng, hand the stage over to Daifeng and also this microphone.
All right, thank you. All right, thank you so much. Thank you. So how should I move forward? Here.
Anywhere on the slide. Yeah.
Oh, okay, that's great. Or just hold it. Yeah, thank you so much. So today I'm Daifeng Wang. I'm a faculty member in BMI. So today I'm going to briefly introduce my recent work, how we can use multimodal learning to understand the single cell functional genomics in human brains and brain disorders. Okay, yeah, so my research actually aims to develop machine learning approaches and bioinformatics tools to open the black box in human brain disorders and try to understand underlying biology in the black box. So at a high level, you can imagine that we can input your genetic information like DNA into some machine learning approaches to improve disease diagnosis, and at the same time try to understand the underlying cellular and molecular mechanisms. So that's why actually machine learning is pretty useful nowadays, because we have generated a lot of multimodal data. So if you look at the population data, so for instance, like for individual people, we can get some phenotypic information like disease type or clinical phenotype. And also now people can measure some like multimodal data at the individual level. So with some, like for example, like genetic variants, like DNA mutation, gene expression, epigenomics, proteomics, and metabolism. So those multimodal data, or it's called the multi-omics data, can be used to improve the phenotype prediction. So if you imagine that if we want to model this problem as the machine learning problem, so basically we have a multimodal data x1, 2, 3. then we try to predict the phenotype or disease type Y. So at the end of the day, the machine learning try to learn some predictive function F to predict Y from X. But the interesting thing is that for the biology, we may have a lot of prior knowledge to see, we know maybe some genes work together in particular conditions. So we can use this prior knowledge to regularize the predictive model. So once we learn this optimal predictive model, so then we can try to use the model to generate new knowledge. Maybe, we found some new genes work together for a particular disease. So this is called interpretability. So this is good, but still a lot of challenges we have to face. So you can imagine those multimodal data are pretty heterogeneous. So they change at different scales, though they may have different distributions. So it's pretty challenging to integrate those data. Also, how we can use those data to improve the disease prediction, and also all the clinical phenotype prediction. Because one of my research focus is about Alzheimer's disease. So how can we integrate this data to improve prediction of a neuropathology or even like a cognitive impairment? And also one challenging also, as I just said, it's biological interpretability. So basically, we don't want to just learn black box model, right? So we try to use the machine learning to infer maybe novel disease genes, or how the gene work together as a gene regulatory networks, or even like how like different cells communicate to each other. So that's a multimodal data at the population level. So, but if we zoom in actually from the individual to the single cells, right now for the individual cells, we even can have multimodal data. So people have actually generated a lot of multimodal data to characterize the single cell functions. For instance, like single neurons. So people actually have generated the data, like gene expression data, epigenomic data, like chromatin accessibility, or even like electrical physiological feature to see how the cells, neuronal cells, corresponds to the external stimulus. So now the challenge is how to integrate those data to find like a cell types or a particular cell cellular functions. So this is the multimodal data for single cells. So actually, recently my lab have published several works about the single cell multimodal learning. So we can summarize those works together as a workflow. So we call it the single cell multimodal learning workflow. So including all the way from the theoretical framework to the data integration, the multimodal data integration, and the cross-modality imputation. Like we can use that as the generative model to generate or impute one modality from the other, and eventually translate our multimodal learning models to the disease prediction and prioritize the disease genes. So today I'm just introduce those work at a very high level. So I'm happy to follow up some technical details if you are interested. So first, let's look at how we can use some theoretical framework to integrate multimodal data. So here it's a URM, like empirical risk minimization for the single modality. So you can imagine that basically, for example, if you try to learn some predictive function to infer some like a handwritten digit, right? So basically you just input a lot of training data and try to learn the optimal function, predictive function between the xi and the yi as the I sample, and basically try to minimize the empirical risk. And also you may have some like prior knowledge to regularize your predictive function. So at the end of the day, you minimize the loss function and try to learn such a optimal predictive function. So a few years ago, we extend this empirical minimization to the multimodal data. So we call it a multi-view URM. So idea is like now we have a multimodal data or multi-view data x1 to 3. So we're still trying to learn the predictive function for each modality. So fi, and try to minimize the loss between the fi x and y. Y is a label. And also now, because we have a multimodal data, so we also try to learn some like a relationship between different modalities. So we call it consensus regularization. So try to learn the relationship across views or modalities. And also, at the same time, we can regularize each modality as well. So we call it a complementary regularization. So overall, we try to we can use this multi-view URM to unify all possible multimodal learning approaches. So for instance, one of the multimodal learning is called alignment-based learning. So basically, you can imagine like basically we try to learn some predictive function to project the multimodal data onto the same common latent space. So you can see for here where I just used the protein expression, common access beta, and gene expression as an example. So you project those genes, proteins, or accessible regions onto the same latent space. So then we can use this latent space to quantify the relationship, the pairwise relationship between the features from different modalities. So at the end of the day, we can get maybe use this relationship to uncover some like unseen like a gene to protein or accessible regions. So this is called alignment-based multi-view learning. So now using a framework, so last year we published a method which is called the DeepMindReg. So basically we use a manifold alignment to learn the relationship between the features from different modalities, and then use that relationship to improve the phenotype prediction. So you can see we have two modalities, modality 1 and 2, and we have cells, like for example, the samples, like cells, individual, like a single cell. So if we don't know the relationship between the features from different modalities, so here we do some like a feature alignment first. So basically, we use the two neural networks to align the features from two modalities onto the same latent space. So you can see on the space A, so we can align the features and try to then use the distance or on the latent space to quantify the feature similarities. So eventually we can get cross-modal feature graph or network. Then use this graph or network to regularize the classification model to classify, for example, the cells from disease or cells from the health or different cell types, like blue versus red. So here I just apply this DeepMindReg to many data set. Here I just want to show how the DeepMindReg can improve the classification of the cortical layers of the neuronal cells. So, for example, if we look at the 3,000 neuronal cells in mouse visual cortex, and we have two modalities, like electrophysiological features and gene expression. So we try to use this method to better classify the multi-class classification, the cortical layers of neuronal cells. The cortical layers can tell you the spatial location of the cells. So you can see here it's a cross-modal feature graph we learned from DeepMindReg. It tells you how the genes close to the electrophysiological features, and eventually you can see the DeepMindReg outperforms others in terms of classifying simultaneously classifying the cortical layers of the neuronal cells. So this is a try to learn the multimodal feature relationship and classify the single cells. But what happened if we don't have a multimodal data? Because generating such a multimodal data is still costly and challenging sometimes. So here, this year we published another model, try to impute one modality from the other. So first, so this model is called GEMI. So basically it's based on the joint variation autoencoder. So first, we still use the multimodal data to infer a joint autoencoder model. So you can see, like we still have two modalities, the X and Y. So we're still trying to learn the modality-specific latent space, so LX and LY. So then we try to aggregate these two latent space using the responding cells with the multimodal data. So you can see this blue vector actually it's aggregated latent representations from two modalities. So then we use this aggregated latent representations and the modality-specific latent space to reconstruct original multimodal data or impute reconstruct the multimodal data. So once we train this model, the good things like once we have data with the single modality, so for example, like a group of cells with just a modality X, so then we just input this single modality data into this pre-trained model and try to infer the other modality for those cells. And at the same time, we can use some techniques to like prioritize the features for the cross-modal imputation. So again, let's still look at our example, previous example, try to impute EFIS features from gene expression. So still for this 3,000, using 3,000 neuronal cells in mouse visual cortex. So you can see the imputed value versus measured EFIS values are pretty correlated for this particular cell type NAM5. And if you look at the multiple cell types, you can see imputed value are pretty consistent with the measured values. And also we use SHAP to prioritize the genes for the EFIS features. So this is like GEMI works for the multimodal data, but what happens if we don't have multimodal data at all? So here we develop another method. It's called the C-mode. It's based on the optimal transport. So it's called the cross-modality optimal transport to impute the multimodal data. So the basically idea is like C-mode try to align the two data set using the manifold alignment or other like alignment method user prefer, and project the cells onto the same latent space. Then for the cells with the single modality data, we just try to map, we call it target cells. We map those target cells onto the aligned space. So yeah, we call aligned cells as the source cells. So then once the using the optimal transport. So of the optimal transport, then we can use the KN to infer the other modality of the target cells. Yeah, so this is just one of the application of the C-mode for the developing human brain. So you can see for the cell type marker genes, the C-mode actually infers those cell type marker genes expression pretty well, and also the highly so basically the genes where can be inferred pretty well by C-mode have corresponding like cell type functions. How many times do I have?
Until 24.
24. Okay, so I try to be fast. So eventually we still want to apply those methods to the disease data. So here, Yeah, so basically, because for the biology, we have some prior knowledge about, for example, gene work together as a gene regulatory network, or how the DNA mutation relate to the genes. So by the QTLs. So basically, as I introduced before, we can use those knowledge to regularize our machine learning model to see if we can improve the disease prediction. So here, we develop another deep learning model, which is called Deep Gummy. So basically, we try to predict the disease type or phenotype, clinical phenotype from the gene expression and the genotype, like DNA mutations. And basically, the idea is like we have a multiple like hidden layers, and the genes are connected by the gene regulatory network. So we call it a biological drop connect layers. So it's not fully connected; it's still connected based on the gene regulatory network. And the DNA mutation like SNP connect to the genes based on the eQTLs. So those are prior knowledge. And then train those network to infer, to predict a phenotype, but at the same time, still try to learn some cross-modality estimation model to learn the latent space between the genes and the SNPs. So this can enable that if you just have a genotype data, so you can still input your genotype data into the model and infer the latent representation of the gene expression, and use the imputed gene expression and the genotype data to finish disease prediction. So yeah, so this is a biological drop connect layers I just introduced. Yeah, this is a loss function. So we tested this method in different scenarios for different diseases, like a schizophrenia versus control, or try to classify different cognitive impairment, or even like Alzheimer progression at different stages. Yeah, here we can show it works pretty well for classifying the schizophrenia, especially for different cell types. And also, after we train the model, we can go back to prioritize the gene regulatory network for the disease prediction. So you can see we can prioritize the gene regulatory network at cell type for different cell types for classifying the schizophrenia. And this is try to predict the cognitive impairment in Alzheimer, and also at the same time prioritize the cell type gene regulatory network for different cognitive impairment in Alzheimer. Yeah, so basically, I just introduced very high level about multimodal data for single cell and how to improve the phenotype prediction. So that's all. Okay, thank you. We have question for
Maybe one question, just a quick one. For follow-up questions, are you going to attend the conference tomorrow morning at all? Maybe we can ask there.
Yeah, send me email first. Thank you.
All right, so thanks for Daifeng again for his excellent talk. And we'll also welcome our second speaker, Zach Huemann. He's currently a graduate student in the physics department, and his current research focuses on using vision language models for disease quantification.
Okay. Thank you for that introduction. You were actually one of my introduction to AI professors, which is funny that it comes full circle like that. But today I'm going to be talking about multimodal learning, in particular, the integration of language into medical image analysis. So first, just a bit about my team. So I'm one of Tyler Bradshaw's students, and I work with Sheen Tai, and then a bit with Junji Hu, who does the NLP side of things. So he's CS and I think biostatistics. But more generally, my research is focused on quantifying metabolic tumor uptake in PET CT scans for lymphoma. So we're really interested in kind of the correlation between how much how much disease burden the patient has and what we can do with that. But what we'll be talking about today is a little bit different. We're going to be talking about a 2D version of that. And that is something called a disease called pneumothorax. So pneumothorax is a disease where you get air between your lung and your chest wall. And so that air kind of stops your lung from being able to expand into that space. And so it's colloquially called like a collapsed lung. And so patients that present with this need to be monitored very closely. And so they get another chest x-ray roughly every 6 hours. So they're repeatedly imaged, and radiologists have to then look back at the images and kind of redo a lot of their work, but to make sure the disease isn't progressing. And so in this work, the kind of workflow we envision is you have this image and you have the previous radiological exam that you've already read. If you can feed it through a multimodal segmentation model, you can get that segmentation out. You can register it to your next exam that you have to do or after, so you have the next exam, so you register them. Now, the segmentation can be overlaid and basically shown this is where the disease was previously, and you can kind of monitor disease volume in that sense. It also can, open up a lot of quantitative approaches, just because you can do like real-time dictation. Like these segmentations take a lot of time for physicians, and so you can speed up workflow. There's all sorts of applications in like central review and different ways in which you have text, you have the images. Can we improve the segmentation given this? And so just to kind of restate this problem statement one more time, because you can kind of see it, you have physician reports that will say there's a small pneumothorax on the right side. Just to note, right corresponds to patient right, not your right. So if I make that mistake, it's always patient right or patient left, so to speak. And then so this is a little bit more descriptive. A right apical pneumothorax. So this is basically telling you, hey, the pneumothorax is at the top of the lung on the right side. And so the way that classical image solutions kind of work is you feed your image through a series of convolutions. And so this is a standard UNet architecture where you basically downsample. And every time you downsample, you're basically doing a max pooling. And so you're decreasing your image resolution. You usually increase the number of channels. But as you come down through this, your image becomes the features become semantically more rich. You're losing individual pixel level information. So then when you decode, you need to put that information, that pixel level information back in, and you do that via these skip connections. And so these skip connections basically take that information and put it back. And you look at an example later on if we have time of that. And so our approach basically takes this UNet and integrates it with large language models and basically inserts this via cross-attention module into this decode side. So as you encode your image features, they come down, and then you have your language features, which are basically word level embeddings for every single word. So they become contextualized by being fed through the language model. And then we project them down using a linear layer and fed through. So they're the query and the value in the cross attention, if you're familiar with the attention is all you need paper. And then those basically give you what comes out of that you can think of as for every word, a weighting. So let's say it might say something like left pneumothorax. It'll basically weight the pixels on the left side differently than the pixels on the right side. And so all of these words basically get learned just through data. We don't do anything else. We just feed it a ton of data and a ton of labels. Actually, it's like 2,000. So it's not that many. And then you're able to basically weight the images according to that and guide the image analysis via this mechanism. And so the tops, that's okay. Whatever. So on the left three, we have 3 vision models. And can I
more?
More.
Should be a high. A little bit lower. I and or the other one. Sorry.
Oh.
Click it again. I'm uploading. Yeah.
Thank you. Okay, so on the left, we have 3 vision models. And so to kind of not go into too much detail, we have the UNet we already talked about, the ResNet, which is just that same UNet with a few extra basically encoding layers, a few extra bells and whistles. And then Gloria, which is the same basically vision model. And then they basically do a contrastive loss to learn a joint embedding, similar to the other multimodal talk we just learned or talked about, except it's between the language features and the vision, but they don't actually include the language in the segmentation. And then On the right, we have LVAT, which is another vision language model, and then our contextual net model here. And on the right, basically the target. And so what we have here on the bottom are scores between 0 and 1. So you can think of a score of 1 as a perfect match between the predicted pixels and the target, and the 0 is basically no overlapping positive pixels. And what you can see is that these vision language models do significantly better, particularly at hard cases. So here we have a very small right apical pneumothorax. Vision language models find it. Vision models are completely lost. Again, another example. So these are kind of, that's one way that, you know, this kind of, the language can guide the image analysis, but it can also reduce false positives. So in this fourth case, you see the vision models are all predicting pneumothorax in both the left and the right side. There's no mention in the report of a left pneumothorax, and so the language models don't make that mistake. And so if you actually aggregate over the whole data set, on the bottom, I use too much of the slide space. You can see our scores, and I guess the short of it is contextual net performs about as good as a doctor actually at this particular task. And so that's kind of the high level findings, but we perform some ablative studies here just to kind of tease out what are the effects of every, you know, each individual component. And the first thing we found is that vision augmentations are really, really important. And so we saw like a 3 to 5 point dice improvement by just using pretty rigorous augmentations. Since we saw vision augmentations were really useful, we want to try text augmentation. So we tried like synonym replacement and shuffling the sentences and different things to try to incorporate different kinds of language, and none of them worked, which I think is kind of interesting, because if anyone has any ideas on how to augment text in a way that, you know, you can create some problems doing it, especially in multimodal, because you have to, you augment the vision, the image, right? You need to make sure the text is still in agreement with that, you know, augmentation. And so there's a lot of, or if you augment the text, your image needs to change also, right? And so there's kind of, you know, some extra difficulties with augmentation in this particular setting. I think third, we found that the model doesn't really matter. So we tried BERT, Roberta large, T5, basically no difference. And then RADBERT, which is Roberta with trained additionally with 4,000 radiology reports, and still basically same score, which I find very interesting that language model doesn't matter in this case. Some stuff on activation functions, basically the negative and the positive that gets preserved by TANCH is optimal in this case. But I think the highest or the most important one is this idea that the point at which you integrate matters. So we basically only include one attention module. So we've got 4 different spots we can integrate. We do at the lowest level and then the highest level. So L4 corresponds to integrating at this lowest point. And what we found is that almost all of our performance was maintained, you know, basically integrating early as opposed to late. So we only saw a very slight decrease. And if we integrate late, right, our performance drops almost back to baseline, actually slightly worse than baseline. I think that's just a little bit of noise, but kind of interesting that you can get almost all your performance by integrating at one point early, and then, you know, letting that basically propagate through. Here's some more kind of analysis we did on this, where we basically swapped, in this first example, we swapped a report that describes a base pneumothorax, so down here, with a report describing an apical pneumothorax. And so the text describes an apical pneumothorax, so it predicts and hallucinates a pneumothorax in the top. Left switch to right, again, you know, you can basically mess with it, and you can, you know, really shows that you can guide the segmentation by the word choices. And this is an interesting one. Large change to smaller. This is still a very large pneumothorax incorporating most of the lung, but the actual pixel volume that's segmented decreases by about 10% in this particular case. So kind of interesting what words end up mattering and what you can kind of do with this. So we ran into a lot of kind of challenges with this and kind of with this direction of study, just because of the medical side of things, right? There's all sorts of HIPAA laws that regulate basically how data stewardship. And so that makes this very hard, because you need text, which basically can, you know, you have to anonymize it, which can be very difficult. You need doctor labels, which are very expensive to acquire. And then you also need images that also, again, fall under HIPAA. And so we only could find one data set that was actually publicly available. And it was only publicly available if you are a a hospital with HIPAA compliant like data stewardship and all of these other hoops we had to jump through, which I think is really interesting because it's almost easier to just build your own data set in this domain than it is to go and find one. And so that was one challenge we ran into. Additionally, I kind of talked about the augmentation of, when you augment the image, making sure the text basically still agrees. So if you flip horizontally, for example, right and left no longer correspond. So we also ran into a lot of computational constraints because not only do you have a vision model, you also have a language model that you also have to manage. And so we actually kind of addressed that by just initially freezing our language model altogether. And that reduces the number of parameters you actually have to backpropagate to. It reduces your graph size if you use PyTorch and whatnot. And so that was kind of how we addressed that, was just basically saying, hey, how about we just don't touch the language model? And so moving forward, we want to do this in PET-CT. So this is basically 3D image scans of patients presenting with lymphoma. And you run into a lot of problems there because we're going to have a lot more pixels, right? 3D versus 2D, right? You add a whole nother dimension, and for every slice, you have a whole nother image. So we expect our voxels to increase by like an order of roughly maybe like a factor of 100. And then also the reports in lymphoma are much longer. And so we kind of imagine this attention mechanism might run into some problems just because you're attending between way more pixels and way more words. And so if you're familiar, right, it's quadratic. And I think the key and the value. So we'll see how that works out, but that's kind of where we're headed. And so we're currently building this data set and kind of working on getting that segmented and kind of moving forward. How much time do I have?
You have until 42.
42. I got one more slide that I think is really cool that I'll just because I have time. So this is basically the actual feature maps. So this is what I was saying, that at a low level, you kind of lose. So this is deep down in the network. You lose a lot of the resolution of the image. So you can't really tell what's going on, but this is still on the decode side. As you come back up, you can see actually there's a lot of structure in the features, which is kind of interesting because in this particular use case, you can actually look at the features and actually get a lot of information. And this is a case where we basically flip again, left to right. And what you can see is that the attention maps actually change quite a bit. And so basically, in this case, there's a left upper zone. It's pushing down on the entire right lung and basically saying this is not pneumothorax. And on the other side, it's kind of leaving some amount on this left upper zone. And if you look at the flipped version, right, the whole left lung is pushed down on, and there's more contrast being created. So you can actually see, in this case, you can actually look at the feature maps and kind of examine them and probe the text and actually see some of the effects of tweaking words and stuff like that. And so it's a very interesting, I think, very fun use case as a grad student, because you can just kind of throw text in and play around and, poke and prod around and see what you can find. So yeah, that's what I got. Are there any questions?
I have one.
Yeah.
I was just wondering how much. So I was just thinking like practical use case, and I imagine that like different radiologists would have different way of describing what they see. I probably there will be no data set where you can actually measure this. But what is your intuition of like? Do you expect to have like different performances for different doctors just by the way that they express? Or are these types of like reports usually very uniform across different radiologists?
So they're definitely not uniform, and I expect that it would probably be better for certain physicians, but it's actually interesting that the labeling physician for this whole data set and the physicians that wrote the reports are actually not the same, which is actually very nice because it actually gives you a little bit more of a robust probe into that, I think, because you're not biased like this physician always says this and he labeled all the data, so then you're only measuring that, right? So I think it's actually pretty robust. It's from the data sets from the University of Auckland, and they had, I want to say the number of actual physicians, like reading physicians, was like 40. So there's 40 physicians that were contributing to the text, but the labeling was done by 6 and primarily by 1. So kind of interesting how that breaks down. But so I expect there would be differences. Yeah. Were there any other questions?
Yeah, could we go back to the slide about the table of accuracy?
Yeah.
Can you explain what's the difference about the tags augmentation and the augmentation?
Sorry, so we're talking about in the vision augmentation. So
I mean, what exactly is the text augmentation?
Oh, what's a text augmentation? Oh, so what we did is we looked at there's these things called ontologies that kind of define a bunch of words, and they give, along with these words and their relationships, synonyms. And so what we did is we looked for words basically in terms that exist within radiology. So we use something called RadLex, and we looked for synonyms between radiological terms. So maybe I'm trying to think of one off the top of my head. Like actually, two similar words that came up earlier is atelectasis and pneumothorax. So atelectasis is a collapsed lung, pneumothorax is slightly different, but it's very similar. And so that could be a hypothetical synonym. So what we would do is we would replace the word pneumothorax maybe with atelectasis in that case some amount of the time. And in my case, I did 15%. And so that's how we basically replace synonyms. Were synonyms that had radiological defined terms additionally. And then sentence shuffle is what you think. We just took the sentences and randomly shuffled it around. The idea being that each sentence would be self-contained and not depend on the previous one very much. And what we found is sentence shuffle didn't affect the score very much. Synonym replacement, a lot. When we did both of them, it's back to not a lot, but basically it doesn't help. That was the key finding. I wouldn't die on the hill saying synonym replacement hurts performance, but I would die on the hill saying it doesn't help, at least in this case. Yeah.
Like our speaker.
All right, so let's welcome our last speaker, Pedro Morgado. So Pedro is also my colleague in the sense that we go to the same conferences. So his research focuses on or lies in the intersection between computer vision and machine learning. And his group develops systems to continuously perceive the world using multisensory data with a recent focus on sort of considering auditory and visual signals. Without further ado, I'll hand this over to Pedro.
All right. So thank you, Lynn, for the introduction. Yeah, so without further ado, I'll just jump right into it. We have 14 minutes. So the talk today is going to be about early fusion and specifically about the benefits of early fusion to learn deeply integrated representations for audiovisual processing tasks. Let's see if I can move on to the next one. Here you go. So audiovisual understanding. So the goal of audiovisual understanding is basically, generally speaking, just to enable computers to understand and interpret multimedia by looking at and analyzing both audio and visual components in your data. More specifically, what I'm usually interested in is in understanding, try to have computers understand nuanced interactions between both the audio and the visual that will enable us to do different tasks from retrieval, like from the audio you want to retrieve the corresponding video or the other way around, or multimodal recognition, meaning from both the audio and the video you want to recognize what are the sound sources that you can see, not just what are they, but where they are, so localization, but also if you have a video where you might have like multiple sound sources, then you may be interested in basically not just localizing the different sound sources, but also separating the sound sources in the audio space and making the connections between the individual sound sources and each one of the individual localized objects. So I've been working on this area for 6 years now. I've been fascinating with audiovisual learning. And one of the things that I found both in my research and in the literature in general is that most of the time people just focus on these late fusion models. Basically, that means that you would process both the visual input and the audio input through unimodal encoders, get some representations, and then because a lot of these tasks you still need to learn interactions between the audio and the video, then they would basically deploy these task-specific decoders where all of the fusion between audio and video would be happening. There is several reasons why this is a good idea. On one hand, you can really, like these unimodal encoders are much easier to train, and you can also already leverage pre-trained foundational models that have already been pre-trained with a lot of data. So you can bootstrap your performance from there. However, for a lot of the tasks that I often I'm interested in, we really need to learn very nuanced interactions between the audio and the visual, and in which case late fusion, it's just not good enough. There's also quite a few compelling evidence for early fusion in human cognition, in human perception. For example, there's this like famous McGurk effect, where basically what you hear is heavily influenced in certain specific situations, but by what you can see. Or there's also other things like the other way around. What you see is influenced by what you can hear. There's also been documented the presence of specific multimodal neurons in the visual cortex that get activated by other modalities, and also like brain regions like the superior temporal sulcus have been recorded to start activity quite early after a multimodal stimuli. Now, yeah, so all of this was just basically a motivation for one paper that I want to introduce today. I'm just going to focus on one single paper. The paper is titled "Masked Autoencoders Are Effective Learners for Audiovisual Early Fusion Models," and this was work done in collaboration with Shentong. Shentong was a master's student when I started working with him at CMU, and is now starting his PhD. And basically, the idea that we want to focus here is how can we develop architectures and the learning methodology in order to effectively use early fusion for audiovisual processing. So there's two questions: the architecture and the training, the learning. The first question is, how can we incorporate early fusion in current architectures, and specifically transformer architectures, just because these days that's what people are interested in and seems to be the way for the future. And how can we use that to learn expressive multimodal representations? One constraint that I really want to incorporate in this work is that we still want to bootstrap from foundational models. So we can think of starting from basically a visual encoder and an audio encoder, in this case, transformers. And then what we're going to do for early fusion is we're going to introduce a set of fusion tokens that the idea is to basically have this set of fusion tokens to be continuously updated throughout the processing stack. And in each one of these fusion blocks, we would be integrating information both from the audio and the visual signals in order to build these integrated multimodal features. Not just that, we also want our fusion representations to be able to influence unimodal processing. So we would also feed back these fusion representations back into the unimodal models. And we can do this because transformers, they don't really depend on the sequence length, so we can just like feed a few more tokens into those models. Now, the key question here is how we're going to really build. What do these fusion blocks really look like? We went through several design steps. The first simplistic approach that we could do was we can just use cross-attention to basically integrate all of the visual tokens and all of the audio tokens and summarize them into our multimodal tokens. So this is a good idea. It's computationally, it's not too expensive. However, one of the things that we're really looking for is representations that can really attend to interactions between specific visual tokens and specific audio tokens. And this cannot do it in just a single step, because we're basically just computing a weight for each token and do a weighted average over all of the visual tokens. Now, on the other hand, what we can do is instead of doing this marginalized integration, what we can do is we start from all of the visual tokens and all of the audio tokens. And then for each pair of tokens, we would compute a representation. And you can think of this as just concatenating the features, a linear projection on top of it. And then, which means that we're going to have a representation for each single pair between audio and visual tokens. And then we're going to update our multimodal tokens by attending to these dense interacted representations. Now, in this case, we are able to extract features from local interactions, but this becomes too heavy because right now we're going to have a very large number of pairwise interactions. What we landed on was basically something in the middle. Instead of attending to all possible interactions, what we're going to do is we're going to first summarize the visual information, all of the visual tokens, into a reduced set of what we call visual aggregation tokens, and we can do this by aggregation. This is the number of aggregation tokens we can specify, and we can make sure that they're small enough. Do the same thing on the audio, and then when we're computing the representations for the interactions, we just compute the interactions between the aggregation tokens. And because we have a smaller number of aggregation tokens, then these pairwise interactions wouldn't be so expensive. And at the same time, we'd still be able to learn information about how the audio and the visual information interact at region levels, basically. Okay, so that was the architecture. Now, what about training? One of the problems of early fusion was basically the fact that because early fusion enhances the how much expressive the model is, these models tend to overfit quite easily. So we really need like a very like a good learning framework in order to basically not overfit. One of the things that actually started this project was just from the observation that mask autoencoders would be like the perfect solution for this task, mostly because they don't really care what is the underlying encoder that you're going to use. So you can use these early fusion autoencoders. And on the other hand, they have already been shown to be very successful for learning representations for individual modalities, specifically for vision and for audio as well. So how does this work? For those of you who may have not seen, we would just start from an image and an audio where the audio is represented in time frequency domain, so STFT. And we both of this, we're going to basically split them into a large number of patches. And what we're going to do is we're just going to throw away a large percentage of these patches and encode the remaining visible patches through our audiovisual early fusion encoder. And then those representations will be fed into the visual decoders and an audio decoder just to basically reconstruct back the patches that we masked out. So basically the loss here would just be an Euclidean distance between the reconstructions and the original patches. So that's all we did for pre-training. We just trained this on a large data set. We use VGG sounds. We had to experiment with a lot of different small changes in the architecture. So for doing that, we used like a fast training schedule of only 100 epochs, which took about 16 hours and on 2 GPUs. Once we were kind of happy with the final formulation and the final architecture, we ran the whole model like with the extended training of 800 epochs, which took about 5 days to train on 8 GPUs. So one of the things that we found right away was that there was 2 specific emergent properties that came out of this training that I was personally surprised by. One is if you already used mask autoencoders, you know that the representations that you learn from mask autoencoders, they're not very good semantically. They do not cluster images very well. However, in this case, the representations that we're learning both for the vision and for the audio were did not provide good semantics still, but the representations that we are obtaining for the fusion representations, for the fusion tokens, those actually were very good at clustering different samples according to their semantics. So here you just see a few of the multimodal retrieval results. Once you feed the base sample, the other samples that would be close to this base sample in the feature space. And you can see that they're actually quite good, despite the fact that we didn't provide any supervision in terms of class labels. The other property was localization. When we looked at the final layer, and specifically the attention map that was computed between the fusion tokens and the individual patches, visual patches, we found that surprisingly, the model was actually looking or searching for those objects more salient in the image, and specifically the objects that were generated the sounds. So, and again, this was doing this despite not providing any explicit supervision. Beyond these emergent properties, we also evaluated our model using 3 tasks. by transferring the weights to downstream tasks. So we use audiovisual recognition, sound source localization, and sound source separation. In all of these cases, we basically transfer the weights that were learned by the mask autoencoder and then fine tune on a small data set with human annotated labels. So not self-supervised, in this case, fully supervised. And the only difference that we had to do is to make sure that we had a small task-specific decoder, either just a simple linear classifier or a UNet for the case of localization. So a task-specific decoder that would allow us to basically provide the predictions that we were interested in. Now, what we saw across all of these different tasks was very consistent across tasks. So here the green triangle is basically telling you the performance. So this is performance versus speed. How many samples, how many frames per second does the model operate on? The green dot, the green triangle is basically telling you the performance of a model that has no fusion. And then what we see with the red dot is that if we provide the ability the model to look at to build early fusion representations, but without the local interactions, then the performance across all tasks improves with a slight decrease in the speed. If we additionally allow the model to look at all of these dense interactions, then the performance improves by quite a lot. This is the blue dot, but the speed gets reduced dramatically. It goes from basically 14 FPS to 4 FPS. But if we allow the model to basically look at factorized interactions that I was talking about, not only we maintain the same performance, but we regain a lot of the speed that we lost before. We also compared this model to a variety of state-of-the-art models on the different tasks, and we show improvements. This was not surprising, given that for the first time we were actually using early fusion representations. And here is just like a quick demo to show the results. So here is just the original video, which there's no sound. Maybe I can just do this. Let me try again. No,
that's the bottom.
Oh, I see. Yep. All right. All right. From this video, you can use the model that was trained for semantic segmentation to identify, in this case, both the piano and the conga. And then once we basically extract the crop just of the piano and feed together with the original audio to do sound source separation, and this is what we get. And if instead we provided the crop of the conga, this would be the separated sound source that you get. There's a few other examples here. I'm not going to go through it. We're already past time. But yeah, yeah. Thank you. If there's any questions, I'll be happy to take them. So this paper, it's a NeurIPS paper. Hopefully it gets in 2 days. If you have papers there, good luck. If you want, it's not on archive yet. If you want a quick pick, just let me know. I'm happy to send the initial version. So yeah, questions?
Sorry to compare things to the brain, but I know that in the brain we have early stages of vision, like AD1, that is completely different.
Yeah.
And not that AI or DNA needs to be modeled off of the brain, but I guess I wondered what you think about that. And is there a middle ground between early fusion and late fusion that people are also testing?
I mean, it's called middle fusion, but no. So I think it's. So I think that's a good point, and I think in this paper we really went all out with early fusion and tried to like integrate from the very, very beginning. And I think if there's enough data and enough compute, the model will be able to learn not to integrate from the very beginning. So like with more data and more compute, that wouldn't be a problem. I think in general, I think a better middle ground is to basically have like a little visual focused stem that is basically only doing vision. The same thing for the audio. But I still believe that, like, basically by the fact that we're mostly using these foundational models that were trained only for vision or only for audio, and then only doing all of the fusion on a small decoder at the top, that is really impeding the progress that you can make on tasks like localizations or separation. Yeah.
All right, good questions. Thanks, Pedro. All right.